The paper I looked at is entitled "The pH Sensing Receptor OGR1 Improves Barrier Function of Epithelial Cells and Inhibits Migration in an Acidic Environment." In the normal human body, our gastrointestinal tract is lined with epithelial cells that contain tight junctions on the apical surface, which prevent bacteria and other unwanted substances from entering the body. On this diagram of the intestinal epithelial cell, the apical surface is where the microvilli are located, and the magenta squares represent tight junctions. Other cell junctions are also labeled, such as the gap junctions and desmosomes, but we won't get into that today. And the figure to the right is an electron micrograph of the cell junctions between the epithelial cells of mice. Various diseases such as Crohn's disease and ulcerative colitis, both of which are inflammatory bowel diseases, affect the epithelial barrier function through the alteration of tight junction protein distribution and expression, which is an ultimate result of the inflammatory response. There is not much current literature about the molecular signaling pathways that contribute to maintaining epithelial barrier function. However, studies have shown that there are 3 G protein-coupled receptors that are able to detect the slight changes in pH from physiological pH, and is therefore considered pH-sensing receptors. In other words, the ligand of these receptors is essentially protons. These receptors are observed to be involved with maintaining homeostasis as well as responding to inflammation and bone metabolism. OGR1, which stands for ovarian cancer G protein coupled receptor 1, is among the 3 pH-sensing receptor proteins. It is expressed in the gut region, more specifically in the epithelium of the intestinal tract. Patients with Crohn's disease and ulcerative colitis have been shown to express this receptor more so than in a control group of patients that do not have inflammatory bowel disease. Previous studies have shown various functions of this particular receptor, including calcium release, PKC activation, as well as serum response factor dependent promoter activity. The researchers in this particular paper specifically investigated the role of OGR1 in human epithelial cell line containing colon adenocarcinoma. They examined the effects of acidic pH on the phenotypic and molecular level. For clarification, acidic pH is a very relative term to physiological pH. For example, acidic could mean a pH of 6. A variety of different methods were used throughout this paper, including cell culture, microarray analysis validated using qPCR, luciferase reporter assay, and confocal. There are a variety of different methods, but we are going to focus on one particular cell culture experiment. To set up their experiment, the investigators used a human epithelial cell line called CACO2, as mentioned before, and then split these cells into 2 groups. They transfected one group of cells with a blank vector, which will become the control group, and then they transfected the other group of cells with a vector plus insert, which in this experiment is the OGR1 gene. That group of cells is the experimental group. Once these group of cells were cultured, each group were further divided into 3 groups, each of which were subjected to a different pH environment, either pH 7.6, pH 7.0, or pH 6.8. All further treatments afterwards were done to cells in each separate pH environment. We will concentrate on the results of a cell culture experiment portion of the paper, specifically the figure that shows the ability of normal cells and overexpressed OGR1 cells to recover after a wound. What the investigators did was take a monolayer of cells of the control group or the overexpressed OGR1 cells and place them over electrodes. Then they measured the impedance in ohms. Impedance is the resistance of the electrical circuit, and So therefore, a higher electrical resistance is indicative of increased barrier function, and a decreased electrical resistance is indicative of decreased cell migration and reorganization. The cellular impedance was measured using electric cell substrate impedance sensing technology, along with automated wound healing or cell migration assays. In the first 6 hours of the experiments, the impedance was measured without affecting the natural course of the cells. But at 6 hours, both groups of cells were subject to wound infliction via a lesion from an electric field. Part A of this figure shows the results of the control group denoted as VC, or vector control, in which the human epithelial cells were transfected with a vector without the OGR1 gene, so essentially transfected with a blank vector. The wounds were introduced at time 6 hours as indicated by the vertical pink dotted line. As you can see, the impedance initially is slightly lower across the pH of 7.6, represented by the black dotted line, pH 7, represented by light pink, and pH 6.8, represented by the magenta line. But overall, there is no significant change in impedance trend over time as the pH decreased. The solid black line represents the mean value, whereas the thick lines of color represent the standard deviation. On the other hand, part B of the figure consists of cells transfected with a vector that contained the OGR1 gene ligated into the vector, thereby overexpressing the OGR1 receptor in the cells. Before any treatment, the pH-dependent impedance increased in acidic pH compared to the impedance of cells in the less acidic environment. This can be observed on the graph as a spike in impedance during hour 1 to 6 of pH 7.0 and 6.8, both represented by pink lines. On the other hand, the blue trend line representing physiological pH shows no increase in impedance. We can therefore see that in acidic pH, the epithelial barrier function increases because the cells are oriented tighter to each other over the electrodes, therefore increasing the resistance of the current that is trying to travel between the cells. And so we observe it as an increased resistance. After the initial wound introduction at hour 6, which is represented by the vertical pink dotted line, the initial impedance decreased across all pH environments and then gradually increased for the first few hours after treatment, which is represented by the small slope or hill on the graph. However, the recovery of impedance was inhibited at more acidic pH. Again, this is pH 7.0 and pH 6.8. In fact, the impedance continuously decreased over time in the more acidic pH environments. However, in the physiological pH, the impedance returned to normal levels over time. Overall, reduced impedance is indicative of inhibited cell migration because during wound healing, cells with damaged barrier function should reorganize and migrate to regain barrier function. And so therefore, in this graph, we should see the impedance rise up again, as we do see with the physiological pH environment. However, that kind of trend is not observed in the cells that are contained in acidic pHs. As a result, acidic pH is also related to decreased cell migration. The study concluded that in an acidic extracellular environment, OGR1 signaling was highly activated, thereby causing signal to transduction cascades that ultimately increased barrier function, as well as upregulated cell adhesion protein and extracellular matrix protein binding genes. As a result, cell migration and growth was inhibited during in vitro wound healing. So my first question is, we've talked in class that pathogens can cause decreased epithelial barrier function, thereby leading to unwanted consequences. So how exactly does increasing barrier function also lead to tissue damage? And then my second question is, since OGR1 is a G protein coupled receptor, can you hypothesize a signal transduction pathway that occurs in the epithelial cell? For example, from when the proton binds to the receptor to the first few couple steps of the pathway. And hint, phospholipids are involved.